Please allow me to introduce today's speaker and one of the co-authors of the book Larone's Medically Important Fungi, A Guide to Identification, Lars Westblade. Dr. Westblade is the director of the Clinical Microbiology Service at New York Presbyterian Weill Cornell Medical Center and an associate professor at Weill Cornell Medicine with appointments in the departments of pathology and laboratory medicine, and pediatrics. We're excited to have him here for this webinar, so without further ado, I'll hand it over to Dr. Westblade. Thank you.
Thank you so much, Megan, and thank you to all of you for joining this afternoon. So here are my disclosures, and here are the objectives of the talk listed. So I hope at the end of this presentation, we'll be able to list yeast species of medical importance, discuss their clinical presentation, and explain diagnostic and antifungal susceptibility characteristics of these yeast species. I also hope that we will continue to celebrate Larone's Medically Important Fungi, A Guide to Identification. So on that note, what I'd like to do is just take us through the anatomy of Larone's before we launch into the talk, if I could. So the beauty of Larone's is such that it was designed by Devies that you really didn't need to know that much about mycology in order to make an identification of an organism recovered in culture. So the first thing you should do is you should start with the Guide to Identification of Fungi in Culture, and you begin to narrow down your organism of interest based on the surface and reverse colony color. And then what you do is that you select the organism line illustration that most closely resembles your organism of interest, and you use the page cross-reference to jump to the detailed description. So in this example, we see Candida parapsilosis, page 130. That's what we're seeing under the microscope. And once we flip to Candida parapsilosis, we see a detailed description. But before that, the new edition of Larone's has actually got color-coded edge tabs that allows you to easily navigate to organism groups. So the yeasts and yeast-like organisms are colored such. When we arrive at that detailed description, we'll see a banner heading which lists the organism name that we as the authors recommend for clinical reporting. There's also a new section, taxonomy notes, that lists recognized names, related species, and species complex members. Macroscopic and microscopic descriptions are provided, and then line illustrations and photomicrographs for added detail. And then finally, there are references for additional information. So the photomicrographs of the yeast and yeast-like organisms that are presented in Larone's are generated using the Dalmu plate method. So I thought what I'd do is just spend one slide describing what the Dalmu plate method is. So this is a method that can be used to differentiate yeast genera and various Candida species because they elaborate different morphologies when using this method. So what you do is you take a young actively growing yeast isolate and you streak it down the center of an area of a cornmeal tween 80 agar plate. You then make 3 to 4 streaks across the first streak to dilute the inoculum. So you then cover that with a glass cover slip, and then you incubate it at room temperature in the dark for 3 days. After that time, you take off the lid of the plate and you examine the cover slip at 100x and 400x magnification. And then what we do is we look at the microscopic images that we see. So here I'm showing an example of what Candida albicans will produce. So we'll see pseudohyphae, true hyphae, budding yeast, and these large terminal chlamydia spores. And then another example is that this is what we can see when we have Candida dublinensis. Again, we'll see pseudohyphae, true hyphae, those blastoconidia or budding yeasts. And instead of seeing a single terminal chlamydia spore, as we do with Candida albicans, we see multiple terminal chlamydia spores. And I often think it looks like a street lamp in Dublin, hence Candida dublinensis. You always want to include a Candida albicans control on that plate. So now I want to move into the meat of the talk, and I thought, how do I really present medically important yeast? And I thought the best way to do that would be through cases. So I'm going to share 3 or 4 cases with you. We'll see how time goes, and hopefully we'll enjoy learning about medically important yeast together through this format. So this is our first case. This is a 79-year-old male with a history of hypertension and chronic lymphocytic leukemia, myelodysplastic syndrome, that had recently progressed to acute myeloid leukemia, AML. He presented to the ED in March 2016 with lethargy, bloody stools, intermittent right lower quadrant abdominal pain, fever, and hypertension. CT showed intussusception, and he was taken to the operating theater for colectomy. And that was hospital day 1. His blood cultures were positive for E. coli, and meropenem was initiated. The patient was in the ICU until hospital day 12. His hospital course was complicated by acute kidney failure requiring hemodialysis and persistent neutropenia and pancytopenia due to his AML. Day 22, an exam was notable for oral thrush and blood cultures are positive for yeast, so mycofungum was initiated. Day 24, blood cultures were positive for VRE, linezolid was started. Day 26, the patient's vital signs worsened and the patient unfortunately died on hospital day 27. The yeast recovered from the blood culture was identified as a low probability Candida species using a biochemical panel. The isolate was sent to a reference lab for identification and antifungal susceptibility testing because it was recovered from a blood culture. And the results of the antifungal susceptibility testing are tabulated below. So I'm going to ask you, based on the time, the period, so it was 2016, the clinical presentation of the patient, so heavily immunosuppressed, and the susceptibility profile, just want you amongst yourselves and to your friends, maybe rattle off some possible identifications. So I'll just stop for a second and let you do that. So I really want you to take into account the susceptibility profile. Okay, I think so. What I'm gonna when I saw the susceptibility profile, immediately I might be thinking about things like Candida krusei, which we know have elevated or intrinsically resistant to fluconazole, but Candida glabrata, which can also have elevated MICs to fluconazole. But then when we take everything into account, this is in New York City, so this was a case that we had in our laboratory here. Well, one Candida auris, and this one in the United States. Learn a little bit more about Candida auris. Clinical cases were initially described in the United States in 2016. California, Florida, Illinois, New York, and Texas are hubs in the US for Candida auris. It's emerged as a multi-pathogen in 50 countries and all continents. Resistant, all three antifungal classes have been described, but the susceptibility profile of US isolates in this little box. About 85% of isolates resistant, but they're resistant amphotericin, and they're documented resistant to ordinance, but notably the mainstay for auris for treating Candida. Or propensity to form bombs and a remarkable ability with the ability to health abilities. So that's why it's a critical disorder. Surveillance disorder. So like the patient that we described in our case. So I want to tell you a little bit about Candida now and Candida auris. So before I do that, launching the diagnosis, I want to talk about histopathology. And so what I'm showing you here is a line illustration and photomicrographs of Candida in tissue. So the morphology of Candida in tissue, we often see narrow-based round to oval budding yeast cells. Their budding occurs singly in chains or in small loose clusters. Pseudohyphae and true hyphae are present. And on the next slide, I'll tell you what I think the difference between pseudohyphae and true hyphae are. The exception is that Candida glabrata cells don't make any hyphal elements. They're just small yeast, budding yeast cells. We often the GSA, which was all fungi specific, so it was non-fungal, but fungi, and also the stain, where fungi will stain pink to red. So just showing the micrograph, you can see Candida albicans, and we can see the small budding yeast of Candida glabrata. Now, the reality is about Candida auris, we don't really know what it looks like in tissue. I've seen reports where it can present like Candida albicans, but I've seen other reports where it basically seems to suggest it presents like Candida glabrata. So we don't really know, at least I don't know, how Candida auris presents in tissue. So what about the pseudohyphae and true hyphae distinction? So pseudohyphae are really chains of blastoconidia that have elongated and not separated from one another. There are constrictions, notably at the point of septation, at the septa. And pseudohyphal branching only occurs at the site of septation. In contrast, true hyphae have no or very slight constriction at the site of septation. And there is often no septation at the initiation of a branch. So that's the difference between pseudohyphae and true hyphae. What about diagnosis of Candida auris? What about the colony morphology? So it's a rapid grower. The colonies are kind of nondescript on the colored. It grows at a range of temperatures, notably up to 42 degrees Celsius, which could be an important characteristic from differentiating it from other Candida species. Its growth is inhibited by cyclohexamide, so it won't grow on media such as mycocell or mycobiotic agar. It has indistinct colonies on Chromega candida, but on a new formulation of Chromega candida called Chromega agar candida plus, Candida auris colonies are cream to light blue with a blue halo, and we'll see examples of that in a second. What about microscopic morphology? The cells are round to ellipsoidal, they're narrow-based budding, and sometimes, very rarely, you can see rudimentary pseudohyphae. The organism is really only accurately identified using molecular methods such as mass spectrometry or nucleic acid-based methods. And initially, the reason why Candida auris seemed to creep through healthcare facilities is because biochemical systems failed to accurately identify it. And it was often misidentified as members of the Candida hemolunii species complex, to which it's very closely related, also Saccharomyces cerevisiae and Rhodotollia glutinus. So certainly if you were to recover something like Candida hemolunii species complex in culture and identified using a biochemical method, you would want to confirm the identification using a molecular method. And probably the same is true for Saccharomyces cerevisiae, but Rhodotollia, as we'll learn about later, is a pink-colored organism. Here is just a table where I just tabulated some key characteristics that can allow one to differentiate between Candida auris and that closely related Candida hemolunii species complex. So notably, auris will grow at 42 degrees Celsius, is not resistant to cyclohexamide, whereas other members of that complex do not grow at 42 degrees Celsius and cyclohexamide resistant. What about Candida chromega? So this is an essential medium that I think every laboratory has to have. You should plate your clinical specimens directly to this medium, particularly blood cultures. It allows easy assessment of whether a specimen is polymicrobic for different yeast species. It also allows rapid identification of key Candida species. So green colored colonies for Candida albicans, tropical blue colored colonies for Candida tropicalis, and pink dry colonies for Candida krusei. Other yeast species have kind of indistinct colony colors. They can be a range of colors, but never those three colors that I just described. What about Candida chromega candida plus? You're able to identify a number of key species, but also has the added advantage of being able to identify or presumptively identify Candida auris. It has those cream to blue colored colonies surrounded by that blue halo. There are other rare yeast species that can exhibit a similar colony morphology, so the identification should be confirmed using a molecular method, preferably MALDI-TOF or nucleic acid-based sequencing. So what about the susceptibility profiles of important Candida species? I've tabulated them here. We've got albicans, auris, glabrata, krusei, Lusitaniae, parapsilosis, and tropicalis. I just want to highlight Candida auris. Generally, the isolates are considered resistant to fluconazole, but it's not considered intrinsically resistant to fluconazole. As we saw with those US isolates, about 85% of them are resistant to fluconazole. It's susceptible, thankfully, to the echinocandins, and it has that variable susceptibility to amphotericin. And just to remind you that Candida krusei is intrinsically resistant to fluconazole, and glabrata can have variable susceptibility to fluconazole. I will highlight Candida lusitaniae. There are reports that it is intrinsically resistant to some agents, such as amphotericin B, but I don't believe that's the case. But what is very interesting about Candida lusitaniae is that it can readily develop resistance to antifungals whilst the patient is on therapy. So we're going to move on to case 2. This is a 39-year-old previously healthy male from Savannah, Georgia in the United States, presented with a 1-month history of progressive headaches, drowsiness, blurred vision, and photosensitivity. Recently pressure washed houses. MRI revealed a non-communicating obstructive hydrocephalus at the level of the 3rd ventricle. An EVD was placed and CSF specimen was collected. So here's the CSF profile. I'll just point out that the protein is high, very high, and there is an elevated white blood cell count with a predominance of PMNs. The CSF grams say no organisms were seen. There were few PMNs observed. The cryptococcal antigen latex agglutination test on the CSF was negative. A 4th generation HIV-2 test was negative, and bacterial and fungal cultures grew cream-colored mucoid colonies within 3 days. So here I'm showing you the fungal culture. See that those nice mucoid yeast colonies. When we take a wet mount, you can see these very spherical cells, okay, and with narrow base budding. We did some biochemical testing. The urease was positive. The organism formed brown-colored colonies on birdseed agar because the organism was able to produce melanin. The isolate was subcultured to special to medium, I might call it, L-cannabinine glycine bromophenol blue, or CGB agar. The isolate grew on that agar and turned it from a green-yellow color to a cobalt blue color. And here I'm showing you the organism growing on that agar and that color change to that blue color. So again, just amongst yourselves, I'm going to ask you, what is the identification based on everything I presented to you? So I'll just sit back and have a sip of my Coke. Okay, so this was a case of Cryptococcus gaddii, and this is a case that Dr. Eileen Bird, a really good friend, and also a co-author in Larone's, and I put together for JCM, and it was a very interesting case. So what is Cryptococcus gaddii? So it's an environmental encapsulated Basidiomycetes yeast that can infect both humans and animals. And notably, I'm Australian, despite my accent might reveal that or not. Maybe I do sound Australian to you, but I always find it interesting that koalas seem particularly susceptible to Cryptococcus gaddii. It's associated with trees, soil, decaying organic matter, including eucalyptus trees traditionally, and also Douglas fir. That was the source of the outbreak in Vancouver Island that occurred in 1999. But I think over 50 different species of trees have been associated with Cryptococcus gaddii now. In contrast, Cryptococcus neoformans is associated with pigeon poop. It's a complex of 5 phenotypically indistinguishable species, hence it's termed a species complex. And I've listed those species here. Cryptococcus gaddii sensu strictu is one of those species. But for clinical reporting, I recommend either calling it Cryptococcus gaddii or probably more formally Cryptococcus gaddii species complex, rather than, if you were to uncover those identifications using molecular methods, reporting them out as the species name. So I think keeping it Cryptococcus gaddii species complex is the way forward. There have been notable Cryptococcus gaddii outbreaks that have occurred in recent history, 1999 in British Columbia and Canada, and also in 2004 in the Pacific Northwest of the United States, particularly in Oregon and Washington. And Cryptococcus gaddii is distributed throughout the United States. So these are data that were shared with me by Sean Lockhart, who again is another wonderful friend and a co-author of Larone's. And so what I want to show you here are 3 species that are part of the Cryptococcus gaddii species complex. And so we've got sensu strictu, Cryptococcus gaddii, Judo gaddii, which is the species that was associated with those outbreaks in the Pacific Northwest and Vancouver Island, and also Basilosporus. So here we can find on the western side of the United States, these 3 species. On the eastern side, we can see that we have a preponderance of gaddii sensu strictu and Basilosporus. And recall that our case is from Georgia, and notably Savannah, which is right down here. So what about infection? You get infected by inhaling basidiospores. The agent of infection is typically related to immune status. So neoformans infects primarily immunocompromised individuals, but it can infect non-immunocompromised individuals as well. Whereas gaddii will kind of, to the best of my knowledge, equally infects immunocompromised and non-immunocompromised individuals. It doesn't seem to have a preponderance for either. It primarily causes, or the 2 organisms primarily cause pneumonia and meningitis. And very interestingly, compared to neoformans, gaddii infection is associated with a higher incidence of cryptococcomas, so these kind of mass-like collections of immune cells in organism. And I'm showing you an image here of one in the brain of an infected patient, huge lesion. The cryptococcomas present in the lung and the brain. It also seems to cause increased neurological morbidity and a slower response to antifungal therapy compared to neoformans. So how do we treat it? Again, I want to remind you that the organism, these 2 organisms, are intrinsically resistant to echinocandins. Asymptomatic infections or mild to moderate pulmonary infections are treated with conosol, so an azole. Severe lung infection or central nervous system infection requires treatment with amphotericin B and 5-flucytosine initially, and then for a long period with fluconazole. What about the histopathology? You cannot differentiate between the two, neoformans or gadii in tissue. The organisms, you see narrow-based budding yeast cells, and this highly variable size of the yeast cells is a characteristic of Cryptococcus in tissue. The cells have thin walls, and some of them can actually resemble, I think they can resemble crushed ping pong balls. And often the cells aren't touching each other because the capsules pushing apart from them and really pushing the tissue apart from them. So you'll see this kind of clearing around the yeast cells. But sometimes the capsule production isn't so pronounced, so that clearing around the cell is not so pronounced. So can we use stains to help us diagnose Cryptococcus neoformans gadii infection? So we can use so-called special stains. So here I'm showing you a lymph node, and I really want to thank my friend who's a master histopathologist, Rebecca Larone, for sharing these slides with me. So here I'm showing you a H&E image, hematoxylin ineosine stained tissue. It's a lymph node, and also lymph node stained with GMS. And so we can see these kind of little soapy-like structures here. Looks like foam of soap. That's the Cryptococcus gadii cells with a clearing around them. GMS binds to fungi and they stain dark black. So what you're seeing here, the dark blackly staining yeast cells, and some of them look like crushed ping pong balls. And we have special stains that we can use to light up Cryptococcus neoformans gadii specifically. We have mucicarmine, which binds to the capsule of Cryptococcus and stains it a bright red color. And here's a really nice example right here. And then we have Fontana Masson, which binds to melanin and will stain the cells black. And if you recall, Cryptococcus neoformans and gadii can make melanin. They contain phenyl oxidase in the cell wall, which produces melanin. So Fontana Masson will light them up. What about lab diagnosis? Direct specimen examination, so gram stain, calcifor white, India ink. Here I'm showing you an example of a gram stain. See these nice spherical cells. Notice they're not touching one another because that capsule is pushing them apart. It's a fungus, contains chitin in the cell wall, so it will light up with calcifor white. And this is the one that we all love to see, but we rarely do anymore, the India ink. Get a CSF specimen, we'll flood the wet mount with India ink, and you'll see that clearing around the yeast cell due to the capsule not being able to preventing the India ink from abutting up against the yeast cell. The mainstay for diagnosing cryptococcosis and a revolution in the diagnosis of cryptococcosis are these cryptococcal antigen tests that detect capsular polysaccharide. They come in two flavors, a latex agglutination test and a lateral flow immunoassay. The tests are performed on serum and CSF. They have very good clinical sensitivity for diagnosing pneumonia and for diagnosing meningitis. But neither microscopy nor cryptococcal antigen tests can differentiate between neoformans and gadii. So how can we do that if we want to? I'll tell you in a little second. But again, when we recover neoformans and gadii in culture, what does the colony look like? It's a rapid grower. It's those mucoid colonies. It's initially cream colored, it later becomes tan, and it can grow at both 25 and 37 degrees Celsius. Here I'm showing you Cryptococcus neoformans / gadii growing on birdseed agar. You see these brown color colonies due to the reduction of melanin. Microscopic morphology, again, using the Dalmu plate method, we're seeing round spherical cells, narrow base budding. Again, just to reiterate it one more time, often the cells aren't touching each other because that capsule is preventing them from touching one another. So you'll see spaces between the cells. Biochemical tests, they're both urease positive. They're melanin positive because of that phenyl oxidase. They can only be differentiated using molecular methods such as mass spectrometry or nucleic acid-based methods, but also they can be differentiated using a phenotypic method. So this is one of my favorite media of all time, CGB agar that I alluded to. So how does it work? So essentially it allows differentiation between neoformans and gadii. Gadii can grow on CGB agar, and once it grows on that agar, it produces a color change from a yellow-green background to a deep blue color or a cobalt blue color. Neoformans will not grow on that medium, and if it does grow on that medium, it usually doesn't elicit a color change. So what's the selectivity in this medium? It's the folks who came up with this medium are brilliant. They exploited the biology of gadii. So gadii loves plants, decaying plant material. So L-cannabinine is an amino acid that's found in certain leguminous plants. It's not inhibited by it, whereas neoformans is inhibited by it. And then once Gattii grows on that medium, it will use glycine as a sole carbon and nitrogen source, and that will lead to a pH increase and ultimately a color change in the medium, so that yellow-green to a blue color. You should only test isolates on this medium that are positive for melanin production, so birdseed agar or caffeic acid disc positive. You shouldn't test melanin negative yeast isolates on this medium because they can elicit a false positive reaction. And you can also generate a false positive reaction with neoformans if your inoculum is too heavy. So you want to use a light inoculum. Most strains will turn positive, and Gattii will turn positive in 2 days, but you may have to hold the media up to 5 days in order to see a positive result. So urease, isn't it such a beautiful test, biochemical test? So again, I just want to ask you amongst yourselves, what other yeasts are urease positive? And I'll just sit back for a second. I'm going to have another sip of my Coke. And okay, well, this is my list. Hopefully it's inclusive. Krusei, reaction may be weak. Lipolytica, Cryptococcus, Neoforms gattii, Malassezia species, Rhodotolula, Sporobolomyces salamonica, and Trichosporin species. So what about case 2 follow-up? Our isolate was ultimately identified using molecular methods at the CDC as Cryptococcus gattii sensu stricto. The patient was presumed to have acquired it through aerosolization of the organism whilst pressure washing houses. The cryptococcal antigen was presumably negative due to the post-zone effects, and we're going to talk about that in a second. So my plea to you all is that if you as providers or as you as laboratorians suspect cryptococcosis in the setting of an initial negative cryptococcal antigen result, dilute the specimen. Dilution is the solution, as I always tell myself. Our patient was treated with amphotericin B and fluconazole, multiple negative CSF cultures, but the protein remained high, and imaging showed worsening ventriculitis. 5-flucytosine and steroids were added, the protein levels decreased, and then a shunt was placed. Patient discharged in hospital day 40, maintained on oral fluconazole. He remained shunt dependent, required 3 shunt revisions over the next 4 months, following intra-abdominal infection with coag-negative staph and 2 episodes of shunt malfunction. The patient expired 9 months after initial hospitalization, probably a result of a shunt blockage. So again, a very unfortunate case. So what about that post-zone effect? And we'll talk about post-zone in a second. So again, this is a real-world example that we had in our lab. And I want to acknowledge Renato Abdel, who took these pictures for me. So here we have the initial test. It's a 1:2 dilution of the CSF specimen. It's the lateral flow immunoassay. We've got a control line that has to be present in order to render the test valid. And then we have where a test line would develop if it was positive. And we don't see that test line there. But the provider felt very strongly that this could be cryptococcosis and asked us to do the dilution is the solution. And so once we did that, here's the dilution series here, increasingly diluting out. I hope you appreciate that you start to see that test line to appear. So this was a classic post-zone, often erroneously called pro-zone effect. So pro-zone is the absence of that antigen antibody precipitation in the presence of excess antibody. But this isn't a test that detects antibody, it detects antigen. So this is really post-zone, where you have an absence of an antigen-antibody complex in the presence of excess antigen. And so when you get everything just right, it's called the zone of equivalence. So just want to remind you, when you're referring to cryptococcal antigen test, it's post-zone, not pro-zone. So we're going to move on to a third case now. I think we're going to have time, Megan, to talk about our 3 cases, so I'm excited. So this is a 54-year-old woman who presented with a 3-week history of fatigue and anorexia. Past medical history was significant for Staph aureus aortic valve endocarditis, requiring valve replacement 9 years prior, and end-stage renal disease following failed renal transplantation. Patient was on prednisone. An electrocardiogram revealed bradycardia with complete heart block. Patient was afebrile, bradycardic, with a pulse rate in the 30s. The patient had 4 peripheral blood cultures drawn on the day of admission that grew Staph epidermidis, so a coagulase negative Staph species. Vancomycin, gentamicin, and rifampin were initiated. A transesophageal echocardiogram demonstrated a thickening of the aortic valve and also suggestion of an abscess. It also revealed a vegetation on the ventricular side of the aortic valve leaflets. On hospital day 2, 3 peripheral blood culture sets were drawn. On day 7, so the 5th day of blood culture incubation, so this was the last day that the blood cultures would be held because they were routine blood cultures. One of the aerobic bottle of one set was positive for yeast. All other blood cultures, including the isolated tubes, which are specifically designed for fungal blood cultures, were negative. On hospital day 7, the patient started empirically on mecafungin. The yeast grew rapidly in culture at 30 and 37 degrees Celsius, rendering these pink color colonies. Again, so what is your identification? I'm showing you the gram stain, the blood culture gram stain. We've got some narrow-base budding yeast, and we've got maybe a rudimentary pseudohyphae. So not too much there. We're not seeing loads of pseudohyphae and true hyphae, so I think we could kick out things like albicans and some of the other Candida species. Doesn't look like Cryptococcus, right? It's not that nice round spherical structure, but the colony morphology is very striking and revealing. So very brilliant pink colored colonies on septex. So this ended up being rototolular species, and it was ultimately conferred using molecular methods as Rototolular mucilaginosa. So a little bit about rototolular. So this is again an environmental organism. It's pigmented due to carotenoid production. It's a Basidiomyces yeast. Its clinically encountered species include those that are listed here. One of the species, notably, has been reclassified into a different genus, Cystobasidium. What about infection? So members of this genus are commonly considered contaminants, but they are increasingly being the cause of severe infections, particularly in immunosuppressed patients. So endocarditis, as with our patient, meningitis, peritonitis. And it's important to remember that this group of yeasts is intrinsically resistant to fluconazole and echinocandins. Laboratory diagnosis, the colony morphology, the growth rate is rapid. The colonies are sometimes mucoid. They're pink to coral colored. Microscopic morphology, again, using the Dalmu plate method, round, spherical, oval, or elongate cells, narrow-base budding, and rudimentary pseudohyphae, as you saw in the example in the gram stain. Biochemical tests, it's urease positive. Remember, it's one of our urease positive yeasts. And I hope you recall that I told you that Candida auris can be misidentified as rototolular glutinous using biochemical platforms, but we can very easily exclude it because Candida auris is cream-colored colony, rototolular is a pink or a coral, Candida auris is a negative urease reaction, rototolular is a positive urease reaction. What about the case follow-up here? So on hospital day 10, after the identification, was confirmed as Rototolular mucilaginosa. Antifungal therapy was changed to liposomal amphotericin B. The patient underwent successful pacemaker implantation and aortic valve replacement. Vegetations were noted intraoperatively. Histopathology of the resected valvular tissue revealed these round to oval yeast and gram-positive cocci. So again, showing you an image, a photomicrograph of GMS stained tissue. The background is green, and you can see these kind of black staining dots, narrow-base budding yeast. In the Browns and hops gram stain, and I have to admit to you all, I'm terrible at reading tissue gram stains. And so again, that's when I call on my friends like Gary Prokop, Bobby Pritt, and Rebecca Marrero-Rolon. And so what I hope you can appreciate here are these pink-colored narrow-base budding yeast, and then also these GPC. Okay. Follow-up, bacterial and fungal cultures from the aortic valve tissue and a hemodialysis catheter with no growth. The patient completed a 6-week course of liposomal amphotericin B, as well as vancomycin, gentamicin, rifampin. Patient was discharged home on hospital day 55. At the time of the report, the patient was maintained on chronic voriconazole suppression and tolerated over a year without recurrence. And so this was a very nice case that was written up before my time, but it was associated with this lab. And so this was written up, a wonderful case by Audrey Schutz and by Matthew Simon, who were in this lab. So a little bit more about pink-colored yeasts. We got rototolular. We see these kind of narrow-based budding yeast. They're round to oval, elongate. What about another pink-colored yeast? It's my all-time favorite yeast. Yeast, I think so. One and only pun today. This is Sporobolomyces salamonica. And again, it's pink-colored, but I would suggest that the hue of pink is different. And you'll also notice these kind of little satellite colonies. That's due to the fact that this organism produces ballistoconidia, not blastoconidia, ballistoconidia, where they're forcibly discharged through these appendages. And the ballistoconidia are these kind of kidney bean-shaped structures. Also, the cells can be very elongated, so up to 35 micrometers in size. So I think those two differentiate one another. And if you really want to do a fun experiment, you have to do the one that I'm going to show you on this on the next slide. And this, in my opinion, is the all-time greatest photograph or image in Larone's. And so this is a method for essentially kind of differentiating pink-colored yeasts. So what you do is you take tape an inoculated corn meal agar plate face to face to an uninoculated agar plate. You incubate it at 25 degrees Celsius with the uninoculated plate facing up for 5 days. And the ballistoconidia will be released from the inoculated plate onto the surface of the uninoculated plate and form a mirror image of the streak on the bottom plate. And Yeasts did this, and I think I just love this image. I'm waiting to get my hands on a Sporobolomyces salamonica so I can do this experiment myself. So we're going to move on to case 4. So we've definitely got time, and I think this is a goodie. This is a very recent one, so I'm trying to keep it contemporary here. And I really want to thank my pediatric infectious diseases colleagues for allowing me to present this. In particular, Melanie Dubois, who was, I think, initially involved in managing this patient. So this is a 12-month-old male with trilateral retinoblastoma, status post 3 cycles of chemotherapy, presents to an outside hospital with fever and neutropenia in December 2023. The patient had MSSA bacteremia that was successfully treated. Patient had a line, a central venous catheter, and it was decided to remove that line because the patient had this recent case of MSSA bacteremia, but also had one in October of 2023. However, after removal of the catheter, he had a persistent lethargy, and CT imaging revealed multiple brain infarcts, and he was transferred to our institution. Subsequent imaging revealed multiple mycotic aneurysms, for which he underwent coil embolization. Repeat imaging revealed leptomeningeal enhancement followed by hydrocephalus, and a catheter was placed. Lumbar puncture was performed, and CSF specimen was collected. So here's the CSF profile. I've tried to do a good job at pulling out the parameters. So the glucose is a smidge low, the protein is high, and the white blood cell count is high, but again with a preponderance of PMNs. A lateral flow cryptococcal antigen test was performed. The CSF was 1 to 5 and the serum was 1 to 40. And I'm going to show you the lateral flow test in a second. Interestingly, the beta-D-glucan was very positive in the CSF, so greater than 3,500 picograms per mil in the CSF, and certainly elevated in the serum. I think the cutoff is greater than 80 picograms per mil for positivity. CSF gram stain, no organism seen. A few PMNs were observed, cultures no growth. So that's where we are. And this is a very recent case that both Rebecca and I were managing with infectious diseases over the holiday period. So here's our cryptococcal antigen test results. I want to show you the serum at the top, and then got CSF at the bottom. Now, I want to point something out to you. So this test, any band is a positive. So here's our 1 to 2 diluted specimen. We clearly see a band, and we can see a faint band out to 1 to 40 on the CSF. And then any further, it's negative. Sorry, it's the serum. Forgive me, that's the serum. On the CSF, we see a very faint band at 1 to 2, and it might be hard to appreciate, but you have to trust me that it's there. We see a faint band at 1 to 5. So we call this positive. We call it 1 to 40 on the serum and 1 to 5 on the CSF. So based on that leptomeningeal enhancement and the cryptococcal antigen and beta-D-glucan results, amphotericin and fluconazole was initiated for fungal. Is it cryptococcal meningitis? Given how sick the patient is, broad antibacterial therapy was initiated, and also the patient had a history of MSSA and had hardware in place, through the coil embolization process. And then also Mycobacterium tuberculosis therapy was initiated. But I want you to sit back and think, friends, what do you think about that cryptococcal antigen and beta-D-glucan test? This is real life. This stuff comes up. And I was talking to Melanie on the phone about these results over the holidays. So what do you think? Any thoughts? Well, why don't we start thinking about what yeasts can cross-react with cryptococcal antigen tests? And again, just for fun, I'll just wait a couple of seconds and see if you want to chip in any results. Okay, all right. Well, Trachosporin species and Rototolula are notable yeasts that can cross-react with cryptococcal antigen tests. So what did we do? We were fortunate to be able to call upon a test that basically interrogates plasma for microbial cell-free DNA. So when microorganisms die, they release their genetic content. And so that's why it's called cell-free DNA. So there's this company called Carius that will sequence microbial cell-free DNA that is present in plasma. So plasma was obtained from this patient, and we detected Trachosporin incon and also a gram-positive and gram-negative bacteria. So based on that result, and also taken together with the beta-D-glucan result and the cryptococcal antigen, therapy was trained from fluconazole to voriconazole. So voriconazole has much better trachosporin coverage than fluconazole. And I should say the reason why we were so kind of surprised about the cryptococcal antigen result and the beta-D-glucan is that Cryptococcus should not cross-react with beta-D-glucan. So that was a kind of alert for us. There are some rare reports that in the setting of HIV, beta-D-glucan can be detected in the presence of cryptococcal meningitis, but it's really the kind of rare report. So that positive beta-D-glucan with a cryptococcal antigen result was steering us away from Cryptococcus because Cryptococcus shouldn't cross-react or react with the beta-D-glucan assay. So again, just getting back to the story, fluconazole was changed to voriconazole for better trachosporin coverage. Broad-range antibacterial therapy was continued. The TB therapy was stopped. We were also able to send off CSF specimen. Very fortunate to be able to work with Charles Chu very closely at UCSF and his wonderful lab to perform unbiased sequencing on the CSF specimen. And again, only Trachosporin incon was detected. So taking everything together, this was a case of Trachosporin incon meningitis, which is truly fascinating. So the cross-reactive cryptococcal antigen test, the positive beta-D-glucan, which shouldn't be positive Cryptococcus meningitis, and these sequencing-based assays support the identification of T. incon and as the cause of the meningitis in this patient. So what's Trachosporin, or as I like to affectionately call them, Trachomoron? These are Basidiomycetes yeast widely dispersed in the environment. There are a number of species that can be recovered from clinical specimens. It's also important to note that they're normal microbiota in skin, human skin, nails, and mouth. They're increasingly involved in invasive, localized, and disseminated infection. Immunocompromised patients with neutropenia are especially susceptible. I mean, the best treatment for a fungal infection is the humble neutrophil, right? The type and site of infection are somewhat predictive of the involved species. So here I've tabulated that. I won't labor this table with you, but essentially I've tabulated the organism and the infection that they can cause. When we look at Trachosporin incon, it's really associated with white piedra of pubic hairs, and we'll talk about white piedra on the next slide just because it's interesting. And it's occasionally been associated with systemic infections. The one that we recover the most often in our lab, and I think is recovered the most often in most people's labs, is Trachosporin asahi, which causes systemic disease and has a predilection for hematogenous dissemination. It's also the cause of this very curious and very interesting summer type hypersensitivity that is observed in Japan. And I can't help but think that's where the specific epithet comes from, asahi, which I believe means sun or sunrise, in keeping with that summer type presentation. So the treatment is voriconazole. It's intrinsically resistant to echinocandins. This is all trachosporins, but notably Trachosporin asahi appears to be inhibited by amphotericin B, but is resistant to its fungicidal effects. So that's why people really want to switch onto voriconazole to treat trachosporin. So what's white piedra? When you see it, I've never seen a case. I'm very interested to see a case, but basically you see these white to beige nodules that are distributed at irregular intervals along a hair shaft. And when you look closely under a microscope, you'll see this kind of like white amorphous mass encasing the hair shaft. So just for fun, friends, I know it's not a yeast, but what's the etiological agent of black piedra? Just talk amongst yourselves. So it's also known as Piedra hortiae, Piedra hortiae. So what about the histopathology? It can resemble aspergillosis. So the organism grows from a focal point in these parallel radiating fashion. But what differentiates it from aspergillosis is that you'll see pleomorphic yeast cells, pseudohyphae, septate true hyphae, without predominant dichotomous branching. So dichotomous branching is associated with aspergillosis and some other, really aspergillosis. And you'll also see arthrochonidia. It can be difficult to differentiate Trachosporin from Candida if arthrochonidia are not observed, at least in tissue. In tissue. And so you may need molecular or culture-based methods to be definitive. What about laboratory diagnosis in our microlab? Colony morphology, it's a rapid to moderate grower. Colonies that are initially moist, but then become kind of wrinkled or powdery crumb-like as their culture ages. It's initially cream-colored, often darkens to a yellowish gray. Microscopically, again on cornmeal agar, we see blastoconidia, so that budding yeast, pseudohyphae, true hyphae, and then we see arthrochonidia. And so this is an example of an arthrochonidia here. It's basically where the true hyphae have broken up into these structures. They almost look bacillus-like. And so here I'm showing you that lovely line illustration of Trachosporin, and then I'm showing a photomicrograph, and here's an image of the colony. Biochemical test, it's urease positive. It's important to note that an organism that can look very similar to it, colony-wise, but also under the microscope, Geotrichum, is urease negative. As I just explained, it can cross-react to cryptococcal antigen tests. And so just for those of you that are interested in tables, I'm a very interesting human. I like to tabulate things. I've tabulated Trachosporin and Geotrichum. Trachosporin is positive, Geotrichum negative. Trachosporin will produce or elaborate blastoconidia, pseudohyphae, true hyphae, and arthrochonidia, whereas Geotrichum only elaborates true hyphae and arthrochonidia. So the follow-up, so this is an ongoing case. So repeat brain imaging showed development of new mycotic aneurysms and new infarcts. Dexamethasone was initiated. Patients currently on voriconazole monotherapy, and now we're seeing the beta-D-glucan levels decrease, which is wonderful to see. So again, a lot of thanks to my PEDS-ID colleagues for sharing this case and all their hard work that they do with this and all their other patients. So now I just want to spend a couple of slides just celebrating resources. So obviously I want to celebrate Larone's, but I want to celebrate all these other resources. So I'm a big fan of Descriptions of Medical Fungi by Sarah Kidd, Katerina Halliday, and David Ellis. I love the book. I've got it. Here's a photo. This is my collection. This is my mycology collection. I also love Richard Somerville's and Guy Saint-Germain's Identifying Fungi. Who doesn't love Manual of Clinical Microbiology? I don't want to know them if they don't like it. And then I absolutely adore Clinical Microbiology Procedures Handbook. I think this is, oh gosh, for my lab, it's huge in how we basically lean on it to fine tune and develop our procedures. And the mycology section is wonderful. I really enjoy the Atlas of Clinical Fungi. I have it and I lean on it a lot. And I think that any lab that performs mycology needs to have a copy of the Atlas too. And so that's Sabin the Hoog's masterpiece. What about websites? So I want to draw your attention to a website that Sarah Kidd was kind enough to tell me about. This is the Yeasts website. It's brilliant. I love it. I love biochemicals. I love memorizing stupid facts about yeasts and microorganisms, and this website has facilitated all my dreams. So I told you I'm very interesting. So this is a great website that you should lean on for your yeasts. And I'm in love with the Adelaide website. It is probably one of the finest resources of mycology. And so I welcome you all to use these resources for mycology, not just yeasts, but all of mycology. Well, certainly just yeast for this website. Acknowledgements. I have to, and I hope I don't bore people, I'm almost done, but I have to give a huge amount of praise to Devis. She started working on this book before I was born, and I've got street miles on me, so I look pretty old. So she has been involved in this book for so long, and done such an incredible job. She is a fantastic individual, beautiful individual, and a wonderful mycologist. And I often tell people when they get given or asked to do something, give thanks. People don't say thanks enough. People don't recognize when they've been thrown a bone. And I need to acknowledge Devis. She threw me a bone. I'm not a mycologist by training, but I love it. And Devis facilitated and helped me take on the next edition of Larone's. And that brings me to celebrating some remarkable people as well. So Devise, I must celebrate her, but I also want to celebrate Megan, who was incredible in guiding us through Larone, and in fact, really deserves her name on the title. True great friend, Eileen Bird, Sean Lockhart, Gary Prokop, who I got to learn about and meet during this process, and I loved it. And also Thomas Walsh worked with us on the book. Incredible people, and very proud to work with them. And I've become fast friends with Sarah Kidd, and I've really enjoyed being able to shoot ideas by her, ask her questions about mycology. So I really appreciate you as well, Sarah. I've also listed all these other people who are just wonderful. They include the artists who are able to help us with some of the line illustrations for the new book. Also people at New York State, David Pincus, who's a lifelong friend who is a mycology geek, but only in the best way, because now I think I'm a mycology geek, and a really good friend, Rebecca Moreira-Rolon, who's my co-pilot, who is a wonderful histopathologist. And also my micro labs. They are remarkable people, and they're the ones that are going in and doing the day-to-day work of mycology. And that, to that end, I just want to just share an acknowledgement that Eileen penned, and it was so beautiful that we decided to put it into Larone's. And I didn't really get emotional, but this is kind of it's a beautiful, beautiful prose. We acknowledge every medical microbiologist who pursues identification of fungal pathogens from increasingly complex populations of patients. We hope that the descriptions on these pages help you navigate the unique challenges of fungal identification and inspire a sense of wonder for the world of medical mycology. Thank you for your devotion and commitment to the field and the patients you serve. And really, truly, thank you. And at that point, I'm going to stop. I think I've left some time for questions. And
yeah.
Thank you.
Thank you so much, Lars. So we're going to move on to the question and answer portion of the webinar right now. We've had some really great questions come through, and we're going to try to get through as many as possible. So the first one, Lars, I'm going to pitch to you is, do you think in the future, all of those interesting methods like CLB agar, precipitation tests, etc., are not going to be used because everything's going to be identified by MALDI-TOF? This person's scared of their favorite microbiology becoming a MALDI-TOF factory.
Well, that's a very good question, but I think we're certainly going to need cryptococcal antigen tests. So we're always going to need it. So don't worry about that. We're always going to have cryptococcal antigen tests. Now the questions about some of these bespoke agars, or specialty agars, it depends on your resources. You can't put mass spectrometers everywhere, and so you may still need to have or employ those media. So it really depends on your resources and your ability to implement mass spectrometry. Now, maybe not in the case of Cryptococcus, but there are also organisms where moldy isn't always truth, and we often have to lean upon biochemicals to help get truth. So for the biochemical aficionados like myself, and clearly this person, I think there's still going to be some room for biochemicals, probably just not as much as we've leaned on before, and that's also a blessing, right? Because it's basically freed up labs in this kind of resource limiting world in both staffing and reagents that we can lean on moldy and do away with a lot of the biochemicals.
All right, next question we have here. So since you mentioned isolator tubes, and I think the third case, given the discontinuation of isolator tubes that can be used for fungal blood culture, how should laboratories perform fungal blood cultures?
Yeah, no, it's a great question. I see the buzzing on DivC and ClinMicroNet. Well, the first thing I'll say is that routine blood cultures, so the ones that we typically collect for bacterial bloodstream infections, do admirably with Candida species, and I think even Cryptococcus. So if you're worried about cryptococcosis or candidemia, then a routine blood culture will suffice. When you start thinking about some of the kind of stranger yeast species, maybe Trachosporin, and certainly some of the filamentous molds, such as Histoplasma, dimorphic molds, Histoplasma, so yeast in the beast, and Scedosporium, you want to try and use some type of method that will pull out these organisms in blood culture. To the best of my knowledge, the only methodology that's available is the myco-ephylytic bottle that's associated with the BD Bactec FX system, or BD Bactec system. So when the isolated tubes were discontinued in my lab, we moved over to the mycoathletic bottles for things like histoplasma, for scedosporium, for fusarium. Just remind you that you will hardly ever recover, if ever, aspergillus or mucormycetes in blood cultures. They're angioinvasive, but we just don't detect them in blood cultures. So really, it's down to the mycoathletic bottle. I'd love to hear from other folks what tricks they've come up with.
Thank you. All right, can you speak about the role of the loss of normal microbiota and high dose and variable bacterial antibiotics in fostering opportunistic yeast infections?
Oh, wow. Shoots, I probably can't speak with any authority, but what I think you're describing here is dysbiosis. And so if you have a happy microbiota environment and then you just challenge it or pulse it with antimicrobials and you affect it, you can lead to overgrowth by other members of that community that could change the environment. So you often get, we call that dysbiosis. We see that most often in patients who are neutropenic, who are being treated with antimicrobials. We can see that their gut microbiota can be greatly affected, and that can lead to gut dominance with a predominant organism, and that could potentially lead as a prelude to disseminated infections. But I can't speak too much more than that. Well, certainly wouldn't want to, because I'm probably not the right person.
All right, moving on. For Candida auris, the CDC's standard recommended culture-based method is still salt dositol. With Candida auris on the rise, we're being asked to stand up testing, and we'd like to use culture versus PCR approach. Are there any studies or anecdotal evidence you know of comparing a Chromagger versus salt dositol approach?
I think the literature you need to look at is comes out of New York State Wadsworth, so Sudha's lab. I think she has a beautiful paper, I know that has a beautiful paper that examines the performance of Chromagar for detecting Candida auris from clinical specimens. And so in my lab, at least, what we do is we collect a swab. It's 4 sites, 1 swab. It's an e-swab. And then we plate some of the e-swab juice to Candida Chromagar, and we basically incubate it for up to a week at 35 degrees Celsius. And basically we ignore colonies that look like albicans, tropicalis, or Krusei, and we send those other colonies to mass spectrometry for identification. We're in the process of validating the Chromagar Candida Plus. So short answer is, I think that you can use an e-swab to collect 4 sites, 1 swab, and you can plate the Chromagar and hold it for a week. And if you want reinforcement, I think you should check out Sudha's paper.
Since you showed so many great different types of resources in this, are there any avenues for proficiency training on mold identification that you can speak on?
Yeah, I will say that Sean Lockhart and his wonderful colleagues at the CDC, I think every 2 years, in collaboration with APHL, have a virtual mold identification course that is absolutely mind-blowingly brilliant. It's virtual, and so that means that people can access it readily. And then I think that Sean Zhang and Saiben Dahoog every 2 years have a kind of in-person mold identification course where a select group of people actually go to a central laboratory, and I think there's lectures in the morning and then there's practicals in the afternoon. So there's 2 great resources for identifying molds. I'll also say, if I can, without overly emphasizing it, I think the Adelaide website has a bunch of unknowns, and I often just find myself looking through those unknowns and just kind of reinforcing my understanding of mold and also yeast microbiology.
All right, we're coming up on the hour, so I think I'm gonna pitch you this last question someone just sent in, which is, what should a young future mycologist start with? Any short parting words or advice?
Oh, keep being enthusiastic, and just immerse yourself in it. Mycology is one of these beautiful disciplines where the more you do, the better you get. So I think getting in the lab, working with unknowns, doing your tape preps, working with the organisms, I think is really important. And then engaging people at societies and meetings. Hopefully you can get to ASM, hopefully you can get to regional meetings. reach out and speak to mycologists. And again, ASM has a lot of resources too. So looking through ASM on the resources that are available for mycology as well. So it's the best time to be a mycologist. In fact, it's the best time to be a clinical microbiologist, but particularly a mycologist. We've got The Last of Us that have basically amplified the interest. You listen to talks by Arturo Casadevall talking about fungi being involved with extinction events. It's just a fascinating time to be a mycologist. So enthusiasm and a willingness to learn and get stuck in. How about that?
I think that's great. So we're right here on the hour. So I want to thank Dr. Westblade for today's excellent webinar and his enthusiasm on the subject. And I'd like to thank the audience for all of your thoughtful questions and for taking the time to join us today. As a reminder, a recording of this presentation is going to be emailed to you within a few days' time. And with that, we're going to end out the webinar. Thank you all so much.
Thank you.